let's go over what it is that should have been gotten done. Let's go over our checklist. Yesterday, we covered some of the basics on ATP and what coupling reactions are. I think we ended with coupling reactions, at least in the video we did. So today, you should have, by this point, read the chapter on photosynthesis that I sent to you a week ago, that I told you to read. You should have completed the photosynthesis packet. That should be done. Your questions on the test are coming from that packet. So if you don't know the answers, if you didn't finish the concept map, I would really focus on that concept map. I would focus on the vocabulary words. I would focus on what's happening at each stage. I would focus on what we cover in this video. What is ATP? How does ATP work? That's all in that packet, and that's all testable material, guaranteed. Half the test is going to be photosynthesis, the other half respiration. Very appropriate. A lot of enzymes involved. Just like there's enzymes involved in DNA synthesis and transcription and translation. All these require enzymes. So we went over how enzymes work. Again, and you should make sure you're familiar with that. All right. And finally, today, we're finishing photosynthesis. The fifth thing is, of course, over the weekend, you need to complete cellular respiration. And that means read the chapter. It's like 17 pages on the chapter, 3 days, and complete the questions. Those questions make you think. They're going to be challenging. Make sure that you are thinking your way through those problems and you write in pencil so that we can correct on Monday. Your test, and let me write that very clearly, your test is on Wednesday of next week. That's Wednesday the 22nd. So there's absolutely no question when your test is on this material, all right? And it's going to be multiple choice and essay. It's going to be both. You have to know this stuff. All right. Go on then. Let's hit this stuff. Let's finish it. It should be fairly clear on what we have to do. So, you have a question? What is ATP is the question. I'm going to ask you to read that packet that I gave you. There's a really clear definition. Ask you to look over that video I sent yesterday, the notes that you took yesterday, or you weren't here yesterday, but the notes are that video we talked about, right? So I go in detail about what ATP is. I can't go over it right now. You can see me after school. I'll be here till 4:15. All right, so in any case, this is a membrane, and what's really cool, it's a specific membrane called the thylakoid membrane. So to review a little bit of the stuff of the material that you should have already watched on the video that I sent you at the beginning of photosynthesis, we have leaves, and leaves have these things called chloroplasts, and the chloroplast looks something like this, and then they have these stacks of pancake-looking things that have chlorophyll in them, the pigment chlorophyll, and they have actually a lot more than that. But these stacks, the stack is called a granum. This space here, this outer space here, is called the stroma. And there's an inner space inside. This whole thing, each of the pancake-looking things are called thylakoids. All right, thylakoids. Yes, please, someone turn the light off. Thank you. So what we have is this whole thing is called a chloroplast. And there's these things called granum. And do you really have to know that they're called granum and that this is stroma and then that's thylakoid? In AP biology, certainly, for your end of course exam, this is supposed to give you orientation, supposed to give you some idea of what's going on where and how. Just know that if it can't hurt if you know it. I don't really expect them to test you on the details of chloroplast anatomy, but they could. Would they be allowed to? Sure, it's an expectation. It's part of your curriculum. I just don't know if they have the number of questions that they need to dedicate to this. I think they're going to ask you more functional questions, in other words. All right, so when we're talking about photosynthesis, we're talking about something that's in numerous steps, okay? And there's the first set of reactions we're going to talk about are called the light reactions. And the reason we call them the light reactions is that they require light. So I think that's pretty clear why someone would call that a light reaction. So there's light here and here this is photon. It's called a photon is a packet of light. All right, so you use light twice in this process. You should know that, and by this time I would expect that you do know. Sorry, I'm going to straighten this out a little bit. You should know. And I would expect that you do know, and if you don't know, well, learn it quickly. All right, so the thing I expect you to know is this, that CO2 plus water in the presence of light yields, right, C6H12O6 plus oxygen gas. Now, is this the overall reaction is something that everybody should know? However, it's not. It doesn't all happen at once. It doesn't happen as one big event. It happens in pieces and parts. So one of the very first steps that happens in this reaction, in this process, is that water is taken up by the roots of the plant. Or does everybody know that? Yeah. All right, so water gets taken up by the roots of the plant, and there's water. Do you see it in the process? Yeah. Well, the very first step, and let's call them steps here, is water is broken up into hydrogen ions, 2 of them, and oxygen, right? And the oxygen is going to have a -2, right? So that's what you break water up into. Now, if you do this twice, what do you end up with? I'm gonna do something kind of nutty here. I'm going to go ahead and put these around parentheses. I'm going to say times 2. All right, what do you end up, if we multiply everything times 2, so we have 2 waters, yields 4 H pluses, right? Plus 2 O2 negatives. What do you think is going to happen with those 2 O2 negatives? So what are they going to do with each other, the oxygens? They're not canceled, they would be -4. They wouldn't cancel, right? So what would they do? What would these 2 atoms, oxygen atoms, do? Let's try to remember what we talked about early on in this class and in Mr. DeCepeda's class. You had 2, 4, 5, 6, right? 2, 4, 5, 6. How do these 2 get 8? What do? They bond, right? And they form O2. That's oxygen gas, right? So what you get is oxygen gas. So by doing this twice, you get oxygen gas. Is that clear? Sir. Is that, I need you to know if that's clear, because this is really important, all right? So this is what's happening here. You're taking water, and you take 2 water molecules, actually. You break them up, and you get hydrogen gas and oxygen gas. What happened to these electrons? Where'd they go? Look at them. There it is. They get sucked up into the photosystem. The electron gets excited by the photon, as we discussed in the previous video. The electron gets excited. It's full of energy. All right, but the key is, where did that electron come from? It came from water. It came from the hydrogen, the electrons got ripped off. You have 4 hydrogen ions floating around inside the thylakoid space. Inside the thylakoid, you have hydrogen ions. Outside, you have less. So that's going to be key. Do you understand all that so far? So that's step 1. That's what happens in step 1. You might say step 1 is the photon coming in and juicing up the electron, however you want to look at it. Okay? But the key is that the first step is you take water and you take the electron off. And you use light to do that. You're using light to do that. All right. So that's the first step in photosynthesis. The next step is that now you have, what you have now is an electron that has a lot of energy. And what happens next is we start to transport it in what's called the electron transport chain. You're going to see the electron transport chain in respiration again. So a mitochondria does a very similar thing. The chloroplast does it, but so does the mitochondria. So when you read the chapter on respiration, and therefore it really would have helped if you read the chapter on photosynthesis already. I know a lot of you haven't. And again, it's a shame because you had a whole day off. But the photosynthesis, that being said, respiration, when you read it, you're going to see the electron transport chain again. So understand what the electron transport chain is. Let's look at it. Let's think about what it is. In general, what it is you have this membrane. Now, there's a lot of details. You're going to read a lot of details in both the respiration and the photosynthesis. When we look at the membranes, the thylakoid membrane, when we look at the mitochondrial matrix membrane, you're going to see proteins embedded in there with all kinds of names and all kinds of functions. Why don't you two separate now before it becomes an issue? If I need to move one of you, I can do it. They know who they are. So you have a membrane, and in this membrane we can embed, as you know, we can embed proteins. Now what's cool is that you have these proteins, and I'm not gonna focus on what the name of each of these are. There are different names in the chloroplast than they have in mitochondria. What I want you to understand is the concept. And at the end, so these things, what they're going to do is that they're going to take an electron, that high energy electron that we produced in that first step of photosynthesis, and they're going to pass it from one protein to the other, like a game of hot potato. By the time it gets down here, it's going to be low energy. This is low energy. And we'll talk about what happens next. All right, so we have a low energy electron at the end. But, and maybe I should draw it differently. Let me draw it differently. Let me say every time it passes on, so every time it passes on, it's just going to get lower every time. That's all I want to make sure I point out. So here you have a low energy electron. Well, that energy in a way was used to try to move it. We used it, or the plant used it, to move a hydrogen ion from this side. What's it going to do? What is that electron going to do to this hydrogen ion? This is where the hydrogen ions are made, right? Where water's broken up on this side. So what's this electron going to do to the hydrogen ion? It's going to take it to the other side. It's going to attract it, and so it's going to go through the protein and end up on this side. And every time it moves, what does it do? It brings another electron. It loses energy, that's right, and it also brings another hydrogen ion over. So it's almost like a trap for it is a trap for these hydrogen ions, and this is the concept that's true for both mitochondria and chloroplast for the electron transport chain. As you transport the electron from one carrier to the next, you're pulling hydrogen ions on one side. And so ultimately, what's the result of this? If we take a look at what the result of this would be, this is before or during. But after you've moved them all over, what you're left with is a membrane, right? You still got your proteins in there. You have your electron is there. But what you're left with is a lot of hydrogen ions over here. Does that make sense? A lot of hydrogen ions over here. And only a couple over here, a very few over here, right? So what do you think is going to happen? Now you got a membrane. You have a lot of hydrogen ions over here and only a couple here. What can happen? What do you see here? What do we call this? You should know this, please. Someone tell me so I feel better about myself. Huh? Can I say this to you, Rick? A concentration gradient. In other words, you have a what? You have a high. That's right. Somebody said it. High to low. Thank God someone said it. You have a high and low. So that means we can have what? What can happen since we have high to low? We can go down. But what is that? What do we call that when things move from high to low? Is this water? Diffusion. That's right. So remember, osmosis is for water. So we have it, but you see, the problem is these hydrogen ions can't come through this membrane. They can only. These are one-way doors. So they come up, and so you have all these hydrogen ions over here, but only have a one-way door. So the question is, how are we going to be able to get these hydrogen ions back? How is again, both the mitochondria, when you see electron transport chain in mitochondria and in chloroplast, what you got to be thinking about is these concentration gradients being formed. And now you have this concentration gradient. How are we going to take advantage of that? Well, we have this big old enzyme which multifaceted and has all kinds of little chunks on it like this, and it spins. It actually spins around, and it has a little channel. And what do you think is going to move through that channel? The hydrogen ions is right. And as it spins, what happens is you take, I'm going to go ahead and put it in red because it's so key. You got ADP is going to turn, plus a P is going to turn into what? ATP. Remember that coupling of reactions? Well, that's what we're doing. We're coupling chemical reactions. So as this hydrogens flow through here from high to low, do we have to put any energy into it? No, they're going to go all on their own, right? In fact, this is the source of the energy, right? The source of the energy that we're putting into that's going into the bonds that make ATP is at the hydrogen ion concentration gradient. And so the hydrogen ion concentration gradient the hydrogen ions move, the enzymes move around, and they allow for a phosphate to be added to ATP. And so now you got this crazy, really high energy molecule that we call the currency of the cell, right? That we can then use to couple other reactions to and drive all kinds of chemical reactions in your body. And so that's ATP. That's what's happening in the middle there. So let me go back for a second. All right, so to be clear, we have the light reactions, and right now where we are with that is we split water. We split water, so water went from H2O to oxygen gas, thank you, very good, plus hydrogen ions. And we did that already, right? And we have a membrane, right? And we took and we moved the electrons. This is, again, just a schematic, right? In what we call Photosystem 2. Photosystem 2 comes first. I know it's crazy, but just know it. Photosystem 2 comes first, and what we did is we took those electrons and we moved them from high energy, and they just kind of dropped down. They got lower every time, and it shouldn't drop that low. They got lower every time, and the electron finally ended up here, right, at photosystem 2, the end of photosystem 2. And we had the concentration gradient and the electron transport chain, and we allowed and then ATP synthase made ATP, all that happened, right? So what do we got as products? We have ATP, we have oxygen gas as products so far, we have hydrogen ions as products so far, and what we have is also ATP so far, right? So that's what we've made so far. Now, we need something to come in and take that electron and recharge that electron. Just like when you recharge your battery. But we're not going to plug it into a wall. You're a plant. What are you going to do? The sun. You get sunlight. Very good. So you get a wave of sunlight comes in and interacts with the electron and makes it go do what? Jump right back up, okay? It makes it jump back up to another carrier protein here. And so this system here, this is called photosystem. That's right. I know. I agree. It should be opposite, but whatever. Photosystem 1, I think it might have been how it was discovered or something like that, but so and I didn't spell photosystem correctly. Let me correct that. Photosystem 1, right? All right, so that's fine. Now we got that high energy electron. What do we got to do with it? We got to transport that electron. Something has to take that electron. By the way, if nothing takes this electron, then the whole thing backs up. Do you ever play a game of hot potato? Yeah. Where you kind of pass the potatoes around? It's kind of fun, right? Yeah. And if you have a lot of hot potatoes and people's hands are full, what happens in the game? It stops. The game stops. So as you're passing these electrons down, if you don't get rid of this electron, there's not going to be anything there to accept it, and the whole chain backs up, and you're done. All right. So we got to get rid of this electron, and it's high energy, so we want to take advantage of that energy, and so we have to transport it out of here. We have to get it out of here. So how are we going to do that? And that's the next question. But does everybody understand so far what I'm saying? Is that we're trying to no, didn't work. Okay. We're trying to get rid of this electron. How do we do it? What are we going to do? Does anybody have an idea? Okay, so if you would have read the chapter, again, if somebody would have please read the chapter, please read the chapter. All right, so there's the last electron acceptor here is NADP plus, then takes the electron, right, and becomes NADPH. So this is your final electron acceptor. For the photosystems, for the light reactions. So now you got these two, it actually has 2 electrons in this. So this NADPH actually takes 2 electrons out of there. And now you have ATP. So this is going to be a key part of the situation, of the problem here. What's going to happen is this ATP and this NADPH are both going to go, well, they're not to the same place. Can anybody tell me where they were going to go? The light reactions are done. Where are they going to go?
To the mitochondria.
To the what?
Mitochondria.
Nope. That's a good guess, but no. Again, read the chapter. So we're done. It's done. We're done with the light reactions. Where are we going to go? The dark reactions. What's another word for the dark reactions? The Calvin cycle. Somebody did their reading. So we go to the Calvin cycle. Now the reason the Calvin cycle is called the dark reactions is because you don't need light. That's all. And there's a bunch. Look, when you read this in the book, when you read this about photosynthesis, you read about respiration. You're going to see a lot of fancy names. Somebody said earlier in the day, big names. There's a lot of big names everywhere. I don't want you to worry about those big names. What I want you to worry about is what I'm outlining for you here, and what I will outline in the video that I send you this weekend for respiration, because I'll send you a video on respiration to help you out. No, you asked me if I was going to send a video on the work. Right, on the mystery questions, and I'm not going to do that. But I am going to send you a video on respiration explaining the steps, like I'm doing here, right?
Right.
Okay, so the Calvin cycle is just literally a cycle, and it just goes step by step around, and there's all kinds of questions about how many carbons are in each of the intermediate steps, and do I need you to worry about that? No, I do not. I do not want you to worry about that. You can worry about that when you get to AP Biology. Look, all I need you to do is understand there's this thing called the Calvin cycle, and its job is to do this. Take in CO2. And no, oxygen's already been given out. Where was oxygen given out? Where? Because that is a question I will ask on the test next week. That is a question that can ask you on the end of course exam. Where is oxygen made in photosynthesis? When you break up the water, the first step. You get oxygen gas there. So yeah, the CO2 gets taken in. What do you do with the CO2? What do you make with that? There's a couple steps after that, actually, but ultimately you end up with something. Somebody said the answer, yeah, but you make sugar, glucose, you make C6H12O6. So you're going to take, where are you going to get the H's? From the hydrogen that's floating around, that's fine. The carbon and the oxygen come from CO2. So plants make sugar from air. Plants make sugar from air, sunlight, and water. They're amazing creatures. It's just mind-blowing that a plant just sits there, breathes, and puts together the molecules that we breathe out, that are in the air already, and it just takes it out and makes sugar. And then, of course, we eat it. So this is the thing that's made. This thing can go to all different kinds of places. The plant can use it to make its structures, right? The stuff you're sitting on, those wooden stools you're sitting on, are made of sugar. So that's the body of the plant, right? So it can make structures. It could take these sugars and it can use it for what? Why would it make this sugar? Because it's making its own food. It can eat it, and the way it eats it is cellular respiration, which you're going to read about this weekend. So you're going to read about how do we take this and then break it up into CO2 and food and energy. This is now, you can take this and you can break it up and get ATP out of it. You're going to read about that this weekend, and I'm going to send you a video on it. Where do you get this CO2? Hold on, before you go, please write this down, because we're not going over this anymore. This is it for photosynthesis. Let me be clear. ATP and NADPH come here. This brings, NADPH brings electrons. ATP is necessary to put the energy into, to stimulate coupled reactions to keep this cycle moving. And out, back to the light reactions go ADP and NADP plus. So both of these go back and it cycles around. So that's it for photosynthesis. Make sure you understand it, where all this stuff happens, what it is. Good luck. Read the chapter, do the questions. You asked me if what? I think that's mine, but if you want it, you can have it. All right, that's it. Have a good one.